Hi, everyone. Thank you very much for joining us today. We are here for the webinar, How to Use AI for Classifying Cell Types and Phenotypes in the Cloud, the Live Dead Assay. And we have a featured speaker today who is Dr. Ilya Goldberg. He was recently appointed as our Chief Science Officer. And today's webinar is presented by our company, ViQi. My name is Julie. I'm on the marketing side here, and I am going to kick us off. All right, so to begin, I want to start us off with a few webinar tips. First, if you have any questions, we will be answering them at the end of this webinar. So you'll be able to ask questions at any point during the webinar by using the question mark icon on the right side of your screen, or some people may have a template that shows them just a box that says questions. So either way, you can use that. If you have any audio issues or visual issues during the webinar, sometimes it's as simple as logging out and back into the webinar. Don't worry, we will have a recording at the end. So if you do have to do that, you will have more details to go back to in the future. So to begin, I just want to tell you a little bit about us. We're hosting this free webinar today. The company is ViQi. We provide large-scale image analysis and visualization expertise, in addition to cloud-based software for scientific researchers and developers. When we work with our partners, they work with us to visualize, analyze, annotate, and store over 250 different types of image and data formats. They can do this in one central repository with our system. And we even work with individual files that are over a terabyte in size, so no limits. In addition to that, we work with a lot of companies to automate and scale their unique research workflows through both machine learning and AI. We can not only help build out those workflows, but if our partners have something they already have put together, we can use that existing workflow or analysis and make it work. So no reinventing the wheel. Last but not least, we do a lot of problem solving for complex image and data challenges. Our team is a group of experts who have been developing this infrastructure for over 15 years, stemming from UCSB. And as mentioned, you'll soon learn about Ilya, who's bringing over 20 years of bioimaging, informatics, and AI to the table, which is wonderful. So let's get introducing Dr. Goldberg. Ilya is here and he'll be speaking to you guys today. He has a long career that lies at the intersection of biology, imaging, and AI. He has done things like co-founding a company that developed the first medical device to receive regulatory clearance for using an AI to predict malignancy in lung nodules in CT exams. He led a research group at NIH, National Institute on Aging, which developed machine learning software for image processing in biology and medicine. While at MIT, he co-founded the OME project, which is still used for imaging infrastructure in large image repositories today. And if that isn't enough, he has over 60 peer-reviewed scientific articles from his time at Johns Hopkins, Harvard, MIT, and NIH in subjects like molecular and cell biology, pattern recognition, image informatics, and the basic biology of aging. So Ilya, I'm going to hand it off to you.
Hi, everyone. So thanks, everyone, for attending. What we want to talk about today is kind of an introduction to AI for experimental biologists and non-AI types of people. We are going to have a very simple example, almost the simplest thing you can do with an AI to perform a simple classification test. So we're going to go through the workflow for doing that. And we're going to spend a bit of time talking about how to evaluate how well the AI is performing, so you can tell if it's working for you or not, and the kinds of things to look for when doing experiments, setting up experiments for training AIs. So next.
Okay, great. So with that, we actually are going to start with an audience poll to just see where everyone is at when it comes to AI. You should all be receiving a prompt on your screen with the poll, which you can select your answer. And the question is, are you currently using AI for your research? So please select one of the following. Yes, we use it in our research workflows often. Yes, but we are just getting started. No, but we are talking about implementing it. Or no, we don't know how it works or how it will help us. So I'll give everyone a few more seconds. It looks like almost all of you have responded. Great. Let's see the results. Okay, so 27% of you said yes, we use it in our research workflows often. 29% of you said yes, but we are just getting started. 40% said no, but we were talking about implementing it. And 5% said no, we don't know how it works or how it will help us. What do you think about those results, Ilya?
So that's good because I think that quite a few of you, as we were hoping, were just getting started with AI, and it's good that some of you have started using it. So if the intro stuff seems very basic, then it will shortly end, and we'll move on to other more practical considerations. This is really a series of AIs. The first one where we wanted to do an introduction and present some initial classification problems. The other webinars in this series, so next we were going to talk about in the following webinar on quantitative problems. So a lot of the problems we have in biology are quantitative. They're actually, a lot of them aren't really continuous variables. They're like standard curves, which are not fully continuous, but there are continuous variable problems also, and we'll show how to use AIs to address those. There are other kinds of problems, which is kind of a hybrid between classification and quantitative problems, where you're trying to gauge phenotypic similarities. So like quantitative similarity between phenotypes that would allow you to cluster things, draw dendrograms, etc. And lastly, we'll cover localization, so finding things in images using AIs and using that for segmentation or heat maps, etc. So that's our series, and let's start off with the first one on Intro to AI. So to begin with, so I'm going to use AI and machine learning and even pattern recognition as essentially meaning the same thing. And I'll be using them interchangeably. They really do mean the same thing, and there are different technologies that allow you to use AI or machine learning, etc. So as far as machine learning, there are two main types of learning. It's important to distinguish them at this point because one of them is much more closely tied to how experiments are done in the lab and how these kinds of AIs would be used in an experimental context. And that's supervised learning. So supervised learning means that you're training an AI on known answers. So you know ahead of time what the answer should be, and you're training an AI to basically come up with the same answer that you already know. And the reason it's a good fit is because that's how controls work in the experimental setup. So if you have a screen, for example, you would have controls for your screen, and those controls are actually what you would train your AI on. And there are other kinds of experiments that fit in that same model, where we know ahead of time what should be happening, and then we have unknowns that we want the AI to work on for us. So then but importantly, we also have unsupervised learning. And the parallel to experimental biology is in unsupervised learning, there are no known endpoints, right? So you don't know the answer ahead of time. And so there are experiments like that. It's not really just doing uncontrolled experiments, although there is reason to do something like that when you're doing exploratory work and you don't really know how what the outcome is going to be, not just for your unknowns, but in general. Do you have a large collection of data and you don't really know how to segregate it? So there are experiments like this. We're not going to do a lot of this because I think um, it doesn't represent what is commonly we're faced with, because commonly we are dealing with controls, and so it's much more better defined, and there is a AI approach to what we most commonly deal with. Lastly, there is semi-supervised learning. So that's where you bootstrap essentially from a trained AI, trained on known outcomes, that you then, for example, for phenotypic similarity, you might start out saying that everything in this is in a separate class, but then you see by measuring how closely related things are to each other that they're in fact clusters and groups that are more closely related than others. And so that is an example of some semi-supervised learning. There are techniques for approaching that. So let's go next. So there are also two different types of technologies for how to train AIs, how to do machine learning. And so I've separated these into two columns here. So deep learning, which a lot of you have heard about, and feature-based AIs. And these are really the two kind of main modern sort of approaches to machine learning and AIs. And I'm sure a lot of you have heard about deep learning. It's very, well, people talk about it all the time. There's a lot of big technology companies, essentially all of them, are involved in this in one way or another. So this is a bit of history of the two things. They've been kind of developed in parallel over the years. Deep learning is based on neural networks. And that's a technology is really the first AI technology. It was invented way back in 1961. And that was the first time that people tried to model, in software, how people thought the brain was working, with simulated neurons connected to each other in software in the simulation, essentially, right? And then people tried to teach those neurons to discriminate things. And so the first learning rule was developed in 1961 by Bernard Woodrow. Then, 10 years later, we have the invention of what essentially is still used as the technique by which you train neural networks and deep learning, convolutional neural networks, et cetera, and that's called backpropagation. And ironically, Paul Warbush invented this, published it in his doctoral thesis, and was forgotten about for 15 years. I mean, this is the most important algorithm in AI still. And so that was rediscovered. And then there was a bunch of work that was done with this. But the big breakthrough really came in 2010 or so, where people started using GPUs to train large networks. That was always the limitation, is to do sophisticated things, you needed large networks and many layers of networks. This is where the deep learning comes from, is that there are many layers of neurons here. And if you have many layers, there are many ways of connecting them. The number of variables that you need to determine becomes exponentially very large, and that's becomes difficult to do. And that's where developments in technology like GPUs really kind of changed our ability to use these kinds of networks. So it wasn't until 2011 or so, a lot of the standard benchmarks were based on recognizing people's handwriting, specifically numbers. As reason was to recognize handwritten zip codes on envelopes for the postal service and for checks, for digital check cashing. So that was the main benchmark. And so the first example of a superhuman recognition, by superhuman, I mean more accurate than an individual person. That was not till 2011. And then the ImageNet was really the first very large set of individual visual recognition kinds of things. And that was essentially the first time that we saw that neural networks, AIs, can recognize a very large collection of very different objects. On the other hand, we have the feature-based AIs. These were started out a little more modern because they weren't based on neural networks from the 60s. And basically, it's a bank of feature algorithms. And I guess I'll go ahead and define that. So feature algorithms are numerical representations of, in our case, image content, right? So if you digest an image down to individual numerical representation of texture or various statistics about the image. So that's where this whole feature idea comes from. And that's been worked on for quite a while. Initially in 2001, Bowen and Murphy at CMU, they developed this thing where they used a set of feature algorithms and a neural network to perform, to identify where a particular immunofluorescence pattern, what subcellular localization it represents. So in other words, identify ER and microtubules and actin and nuclei and lysosomes, endosomes, that kind of thing. There's about 10 or 12 cellular compartments that this AI was able to identify. And the interesting thing is that way back in 2001, Bowen and Murphy showed that this AI could discriminate between endosomes and lysosomes. Endosomes and lysosomes were interesting because no one else could distinguish them. So of course you had resident proteins that you could label, and so you knew what the image was of because you knew what the label was. But visually, there was no way to distinguish them. And the AI could do that with 90% accuracy. And that was really the first indication that AIs could outperform people. So then there's a bunch of developments by Murphy's lab and their software, by my lab, and by Ann Carpenter's lab. we gave us P-SLID from Murphy, WinCharm from my group, and Cell Profiler from Ann Carpenter. And those are still in use today. So let's go on to, so this is just a diagram. I'm not going to go into this in detail, just to give you really a flavor of how the two different kinds of AI technologies work. So in deep learning, really you have, like I said, a large collection of neurons, and they're all represented by these little squares here. And the neurons are connected to each other, just like real neurons. And they perform essentially, this is modeled after the visual cortex. And so it's modeled after how neurons are connected in the visual cortex. And the way that works is that groups of neurons are, so not every neuron is connected to every other neuron. That's called fully connected. And for visual things like deep learning for image analysis, the first layers of this are convolutional. So the idea is that you feed image pixels, and so you get a pixel per neuron at the input layer, and then groups of them are convolved together, and they contact the next layer in the network. And the idea with that is to reduce the pixels, the raw pixels that you feed in, to essentially patterns of, again, image content. And so they're called, in fact, feature maps, because they perform the same function that we do with the other technology, where we use algorithms to extract image content and digest it into numerical representation. So effectively, these are smaller maps that result from the initial input of raw pixels. And eventually those get fed into, typically this is a fully connected network in blue here, where everything's connected to everything. And that's where you have your classifier. And what you do in training with the backpropagation algorithm is you figure out the weights. So each neuron has multiple inputs, and it has a single output, whether it's activated or not, and then that's connected to the next set of neurons in the chain. So each neuron integrates over its inputs, and it has a transfer function, and it either is activated or not, and those inputs have weights. So the backpropagation algorithm figures out what those weights are based on the known outcomes, right? So it has a target for what it's looking at. The 60 thing here, it needs to light up the 60, the neuron that is representing 60 in the output, right? The thing in red. So, and also potentially a category, so it's a sign rather than a bus. So that's essentially how deep learning works, and how individual neurons result in an AI to classify things. So let's move on to feature-based AI. So feature-based AIs are similar in completely different technologies. So now we have algorithms that ingest, again, pixels as their inputs, and then they produce some kind of numerical output, like the average pixel value, or how many clumps of pixels are there, or what is their distribution or average size, or textures. So textures are the spatially coordinated change in pixel values. So it's exactly what you'd think of as a texture. There's all kinds of shape descriptors. So they're all individual algorithm. An algorithm would run on an image or a piece of an image, and it would produce one or more numbers depending on what the algorithm is. And that's feature extraction. You run your image through dozens of these algorithms. So then in the next stage, which again we kind of saw in the neural network layout, is the coordination of these features they're assembling. In this case, we want to figure out which ones are important, which ones aren't, right? And that's kind of equivalent to the weights that we have in the connections between the neurons. And there are different algorithms for that. So these are, again, individual discrete algorithms that give you a score for how good each feature is. And there are specific ones like a Pearson correlation, for instance, would be a good way of determining if a feature is any good for predicting a certain variable, because if it's correlated with it, like with a Pearson correlation, then it's a good predictor. And there are other techniques for scoring how well each individual feature is at solving the problem. And then once that's done and you have weights and a collection of the features that actually ended up being useful, you send them all into a classifier. And those are, again, based on fairly different ideas of how you separate your different classes. And the specific algorithms that are modern that people use these days, there are things called random forests, support vector machines, various distance methods, etc. So in contrast to neural networks, which is all one algorithm, sort of networked together, the algorithm being how you represent a neuron. In this case, we have a lot of different algorithms that each perform different functions, and they're kind of connected to each other, and also in a simplistic way, but resulting in the final answer, which is a prediction based on input data, what kind of predefined class this input data belongs to. So let's go to the last slide of comparing these two things. The two different systems, they're, like I said, both in fairly common use. And I'm not going to go through all of these individually. I'll just say that overall, deep learning, one of the major pros to it is that it's only based on one algorithm. So if you don't have, for instance, a good way of representing numerically image content, you don't need to do that. This becomes starts to play a significant role in multidimensional data. There are plenty of feature extraction algorithms for 1D and 2D data, and even for 3D, things work pretty well. You go beyond that and you start to really have a shortage of ways of representing image content numerically, and that's where deep learning starts to come in. The major shortcoming of deep learning, I think, still is that there's first of all a huge number of parameters. So all of those connections between the neurons, each of them is an individual variable that you need to adjust. So there's a huge parameter space, which is what the training is for. The training is a lot less straightforward. So it requires a lot more data in order to train all of these variables. And if you don't have enough data, one thing that happens often is that these networks tend to get overtrained. So they're too closely matched to the particular inputs that you give them, and then they're not good at predicting real unknowns, data that the network has never seen before, which is less of a problem with feature-based AIs. Typically, you can train them with a lot less data. So let's move on. Of course, you can combine these things. So you can take pieces of the neural network, the feature extraction part, connected to classifiers, and produce hybrid AIs. Also, what works really well is to have multiple AIs using different technologies. right, combined together to give you the final answer. And that way is one way of getting around various biases that are sort of based on that result from the way that specific individual AIs are implemented. I think that's the end of the sort of AI introduction, right? So let's go move on to training. So this is an example of how you would train a very simple AI, which is a binary classifier. And there's a lot of examples of binary classification problems. So we kind of put it in the title, the life-death assay is very sort of common assay to do in cell biology or just biology in general with imaging. But you can generalize that. I mean, a lot of things have positive and negative controls. Well, positive and negative controls are a perfect thing to train an AI on. And it'll tell you, does your thing look more like a positive control or more like your negative control? And an equivalent to that is treated and untreated samples. In clinical work, we have a lot of case control studies. I mean, a lot of medicine is based on case control studies. And that's essentially a binary classification, like benign, malignant. So in the example screenshots that follow, we're going to tag areas of an image and with labels. And then we can tag, or alternatively, we can tag whole images with a particular class. So let's start with the next screenshot. So this is a screenshot from ViQi's platform, and you can do this in a browser where you're presented with an image. Here we're just looking at bright field, but you can see that we have other labels in here that we can turn on and off to allow us to manually by eye label these cells, because we know that they're one thing versus another thing based on our knowledge of the system. And so this is our input into the AI for what we wanted to do eventually and label cells for us, right? So in the next slide, we can alternatively have a conventional image processing algorithm go and segment out all the cells for us, and then we would label them afterwards. And here, the colors are just showing us how the segmentation algorithm is separating cells. So cells that are close to each other, potentially touching, would have different colors assigned to them based on the cell ID, if you will. So those can be labeled also as an alternative path and sent to the AI. And in the next slide, and see, well, what is it that we're actually sending to the AI? So that when you click on cells, or the segmentation algorithm identifies them for you, you can extract a lot of standard sort of descriptors of these cells. There's the image mask, that the individual pixels that comprise the cell. And you can get various kinds of textures, or the perimeter shape or form factors, etc. And you can, those are natural sort of descriptors, numerical descriptors of what these cells are. And so, these are all in all these different columns here. And there, of course, the cell has a representation that we saw in the previous images in the different fluorescence channels. So there's descriptors of the bright field channel, and separately descriptors in all the other channels that you have for the same cell. And they could be the same descriptor, they just apply to the other channels. So what you end up with is this feature table. I think this one is 60 columns wide or so. We're looking at just a piece of it. Also, you see here that's highlighted towards the middle is the class column. So the class column is the other very important input into the AI. And this is called different things. Typically, either the class or the label is also sometimes used as the name for this. This is your target. What are you training the AI to do? You're training it to predict the class, right? And you're training, because this is supervised learning, to do the training, you're giving it which set of columns for a given row correspond to a particular class that you've labeled, right? And so those together are given to the AI. The AIs, the feature-based AIs, they don't care what this numerical content is. This could be categories or floating point numbers or whatever you want, and it doesn't care how many columns you have either. I mean, so really the AI's task at this point is has nothing to do with images. It's simply using all of the columns here, but one, all of the columns other than the class, to predict what the class column would be. And you can actually set the AI to be that general. So given a table, here's the class column, and use all the other columns to predict the class. And AI systems that are fairly automated would go ahead and do that. So that's essentially what you're doing when you're training a feature-based AI. Of course, in deep learning, you would just feed it the pixel mask, and it would figure out all of this numerical content internally. So next, we'll look at, in our case, deep learning AIs are almost there for push button AI. With feature-based AIs, they can't, because the number of parameters that you have is fairly small. You can automate the whole thing and have it run through multiple different algorithms and choose them for you automatically based on parameter searches. And so you can have essentially a very, very simplified way of training the AI. And so you have input data, which is your collection of images that all have the same set of columns and some labels for what you decided what was what. Then you have the other parameters, so that's the input data. Then you have the other parameters, which really just let you say, are you interested in labels on particular objects? So what kind of cell is this in a field of cells? Or are you interested in labels attached to images? So this image is from a well that was treated, and this image is from a well that wasn't treated. So you're basically selecting what tag you're going to use for the training. And that's it. The AI should really do everything else. So you click the run button, and then you get a result, which is in the next slide. That would be, in this case, a labeling of cells by class, right? So now the color coding is by what the AI predicted to be the class. And again, you need to evaluate how well the AI did. Since you manually trained it, you have to have you should have at least a chance to evaluate visually what the AI did and get your impression of whether it's doing a good job or not. But there's obviously numerical ways of checking how well the AI did, and that will go in the next set of slides. So the first thing is a training curve, and that's to answer the question of, is there enough data? And so you can see an example of really successful training on the left, where so notice the scale, first of all, that it goes from 90 to 100. So we're looking at the very top of the scale for performance. So this is the scale on the Y, we're looking at accuracy essentially. So 100% would be a fully accurate classifier, right? And on the bottom, we're looking at the fraction of the samples that we used to get that level of performance. And so you can see as you increase the samples, you get better performance. And then you have kind of a plateau, or at least you have a region of seriously diminishing returns, and it plateaus out at a fairly high level. So we get, with 60 or so % of the samples, we are essentially topped out at just short of 98% accuracy. So that's really good. In contrast, on the right, you have a situation where you have many samples, and you're kind of reached the diminishing return area of AI training, and it's still not great. It's at 60%. And so the answer here is that, yeah, you have enough data, but it's not, for whatever reason, it's not enough to train an AI. The AI is not performing well. Whereas on the left, you've successfully trained your AI, and now you can test it on real unknowns, or actually you want to test it on a withheld data set first. The other thing to look here in the coming slides is the error bars. So you want to make sure that each one of these dots is a separate AI that was trained on the indicated number of samples. And you want to make sure that each time you train an AI, it's based on a subset of your training data, and then it uses the rest of your training data to check it. And so you want to make sure that the sampling is such that you don't have a lot of variability. It looks like a lot of variability with the one on the left, but it's actually very small because the scale is extended. So that's the training curve. Next is parameter sensitivity. So the same idea here, you're sweeping across different kinds of parameters. You want to make sure here that your performance is good, obviously, but that you're not in a sharp little peak, because if you move even a little bit off of a sharp peak, right, your performance would suffer. So that means if you change your parameter by a little bit, you would cause it to your AI to not perform as well. And so here you can see these are sort of all broad regions, and this is support vector machine parameters, and there's only 2 of them. There's actually 3 in the whole 3 parameters for the whole AI training. And so this is telling you that this is a nice stable sort of AI that we've trained here. And next we have the confusion matrix. So this here we're using a different example. This is lilies, I believe. We're classifying lilies based on some input data. And so a confusion matrix, the way you read it is that the known class, so at the top here, Setosa, right, is the known class. And then each column is the predicted class. So Setosa is being predicted as Setosa 13 times out of 13, right? So great. And on the left, we're looking at percentages. I mean, on the right, we're looking at percentages. On the left, we're looking at individual calls, and they're 100% correct. In contrast, for varies color, we have some error with Virginica, right? And so it gets them mostly right, but there's some confusion between versicolor Virginica, right? That's why this is called a confusion matrix, because it lets you identify classes that are confused with other classes. And this poses some questions about how you might want to restructure your AI in order to improve the situation if it needs to be improved. Let's keep going to overtraining. So I talked a little bit about overtraining and how you have to really be careful about it. The message here is you really should withhold a set of data for training once you have a candidate AI that's been trained, right, to test on your withheld data. So in other words, you know all the answers, but the AI has never seen that data before, and you really want to evaluate your AI based on that rather than its internal checks that it will report to you, because those checks are done during training and not with a completely withheld set. And this goes into a little bit of difference in how we talk about validation and testing. So in the AI world, so that's machine learning, AI, deep learning, data science, the validation sets, people typically talk about these internal sets that the AI uses to see how well it's doing internally, right? And the test set is this withheld set that the AI never sees during training that you use to determine how well the final AI is performing. This is actually very different in how validation and testing is used in the regulatory field and possibly in common use also. Validation there is that final set that you use to validate the final AI model, right? The test sets are the ones that you use for internal testing while you're developing your AI. So be very careful about when people talk about validation and testing, what they're talking about, because they mean different things in these fields. So let's go next slide. I wanted to talk a little bit about bias, and that will be the last part of this webinar. So the most common source of bias is disproportionate class sizes. And that's where if you have a training set, 90% of it is class A and 10% of it is class B, a predictor can, if it always guessed class A, no matter what, right, it would be 90% accurate, right? So you have to be, when you're dealing with disproportionate class sizes, you have to be very careful about how your AI is tested and what your accuracy figure actually means. And you have to be careful about how it's trained, because some AIs will actually train themselves to the point and say, well, I always win by guessing class A in this case, so that's what I'm going to do. So you don't want the AI to do this. You want to force it into making an actual decision. And so really there are ways of compensating for this, but if you can, the best way to do this is to force the classes to be equal sizes. Then the other last bit of, we'll talk about bias is on the next slide. These are, your data is often skewed simply, and even if you have equal class sizes, they may be different things are misrepresented that you haven't accounted for. That's sort of the typical bias that you might have in your training data. And it's not enough to just say, well, I have random training data, so it's not biased, because the bias could still be there. There are a couple of things you can actually do. It's not hopeless. So first of all, you can measure the bias. You can sort of say, look, I have a bias variable here. Images were collected by two different postdocs. And, you know, can the AI distinguish between the two postdocs? So you make, you know, instead of treated and untreated, you know, say, you know, Bob and Margaret collected these images. Can the AI distinguish them? If the AI comes back and says, no, I can't, so it has bad performance, then you know it's not sensitive to bias, and so everything's fine, right? On the other hand, if it comes back and says, yes, I can distinguish them, right, then you can still do some things about that. So you can confuse the AI with regards to the bias. So in other words, you have an even distribution between your two postdocs in each class. And so the AI has nothing to latch onto to train itself to just recognize one of your postdocs. And there are other ways of systematically training out this bias, which is kind of more advanced. We're not going to get into, but it is possible to train an AI to not recognize certain bias variables. And I think we're done. I think we have a parting thought here, which is that the performance of your AI ultimately is completely dependent on how accurate your class labels are, and the degree to which your training sets represent reality. And the important thing to keep track of here is that we are dealing, in fact, with tools that can see and detect patterns much better than we can. And so, first of all, when you train these AIs, you don't want to necessarily rely on a single person's idea of what is in this image if you're using image content as your training basis. You might want to get multiple reads or get a consensus read to get a more accurate class label. And of course, you have to be absolutely conscious about bias because these are kind of black boxes. And so they could be latching on and learning from things that you really didn't intend. So that's about all I wanted to say about AI. Sorry if I've gone over a little bit, but hopefully we'll have some questions to answer in the next part.
Yes, that's great. Thank you so much, Ilya. And actually, we're going to do one more quick audience poll before we get to questions. If you've submitted a question and we don't get a chance to get to it, we will follow up with you. But we'll try to fit one or two in after this if you can stick around. So here is a quick audience poll. And it is, do you have an interest outside of using AI for imaging? So you can choose from yes, I'm interested in using AI for genomics. Yes, I'm interested in using AI for proteomics. Yes, I'm interested in using AI for cheminformatics. Yes, I'm interested in using AI at the intersection of one or even more of the above and imaging. Or no, I'm only interested in AI for imaging. So looks like most of you have answered. We're going to close the poll. And here are our results. All right, so we have 3% interested in cheminformatics, 39% at the intersection of one of the above and imaging, and almost 60% of you said, nope, I'm only interested in AI for imaging. Great.
Yeah, it's interesting. It's kind of actually what we're hoping for is that everybody would be interested in AI for imaging and potentially with other things. And so I think we got a little bit of what we were hoping for.
Yeah. Great. Well, thank you guys so much. We are going to take one or two questions right now. If you have any, throw them in. We'll follow up with you after if we don't get to it. And just a quick note, if you need to get in touch with us in the future, feel free to reach out directly at info@viqi.org or visit our website, viqi.org. After this webinar, we do have a quick 4-question survey when you exit the webinar. We'd love to hear your feedback. It helps us provide the best content for you guys. So let's get started. Ilya, we have a question here that says, how much data is enough data to train the AI?
Right, so that's a very common question. And the answer to that, so I specifically, well, maybe you saw a hint that there's how many samples were being used in training. The real answer is that you don't know. You don't know, but there's a way to measure if you got enough, and that's the training curve. So typically, if you have a situation where you can tell the difference between things that you're trying to classify, so in other words, just you looking at them, you can distinguish the different things you're looking for. That's in the for feature-based AIs, it's, you know, a few dozen samples is certainly enough to get started and to see if, you know, where you are to run the experiment, if you will. In cases where you can't tell the difference, you probably, you know, a few dozen is probably not enough. You probably want 100 or more, or even a few hundred samples. But you know, you have tools to tell you whether you have enough data or not. So it's a big range, right? And it really depends on what is, you know, how difficult the imaging problem really is.
Great.
It's not thousands, right? So.
Well, we have another question from Jacob, who says, what are good pieces of software to use for developing neural network and feature-based AI?
So I believe I listed some options for feature-based AIs. So the a big sort of tool that I think everybody in the field uses nowadays is called sklearn. I don't think I put that up there. So that's a Python. So first of all of this is in Python. That seems to be the language that kind of nearly everyone is latched onto for doing this stuff themselves, like programmatically, and using the raw tools to do AI work. If you're looking for sort of clicking kind of stuff, so ViQi has a platform for doing this. Cell Profiler does also. That's just UI based, so there's no programming to do. In terms of deep learning, the reason I didn't identify individual software for this audience is because there really isn't sort of push button AI that uses deep learning. I mean, we're developing that. I believe Ann Carpenter's group is working on that also. I think in the coming year or so, we'll start to see the, you know, initial attempts at this, but it's a lot trickier to put together deep learning AIs, but we're getting there.
Thanks, Ilya. Let's try and fit two more questions in. Demir says, in a feature-based AI, can adding too many features cause poor classification performance?
So the thing is, the short answer is no. And the reason for that is because it has to do with the classifiers. So some classifiers, like if you go from standard statistics, there's the problem of overfitting the data because you've given it too many variables to sort of tune and fit your data very tightly. And so it'll fit your training set really well and then won't be able to predict anything. Well, so that statistics kind of approach to this, where you're just doing fitting or regression or whatever. You know, the way that feature-based AI distinguishes itself is that the classifiers that are used. These are pretty sophisticated nonlinear sort of algorithms that, in fact, are all the modern ones, the ones that I've mentioned. There are obviously ones that are, you know, not used these days because they are sensitive to too many features. But the answer is no. I mean, nowadays, the support vector machines, random forest, some of the distance-based methods, they're totally insensitive to the number of features. And I would say totally within reason. I mean, certainly if you have a factor of 10 more features than are really useful to the AI, it's probably fine. If you're talking about a factor of 100 more features, there might be problems. But actually, it is one of the evaluation tools that's fairly standard. That's just like the learning curve, right? Where you sweep across a % of the samples you're feeding that you're training AI with. Well, number of features is another parameter that you can tune. And in fact, it is tuned automatically in ViQi systems and others where you know, it sort of selects an area where an appropriate number of features. Those curves are very broad. So if you have a dip, it's way out, ridiculous territory. You have thousands and thousands of features.
Great. Thanks, Ilya. And let's take one more question. And if we did not get to your question, we will follow up with you guys afterwards. So final question. This is from Marissa. If I have non-imaging data that I would like to use to train my imaging AI, how would I do that?
Well, so I showed you this feature table, right? So that's just the regular table. It's like a spreadsheet. It is a spreadsheet. And so feature-based AIs, once they digest the image content down to numbers, right? Well, you can insert any columns that you want with numbers that describe your samples in some way that hasn't been extracted from the image. And then you just feed the whole table into the AI. And in fact, the AI will give you feedback on which columns were more important based on these feature selection algorithms, right? It'll tell you, whether the column that you added that had, whatever numerical content, whether it actually was selected in the AI and how big of a role it played compared to the others. So yeah, that's absolutely, it's a little bit harder to do with deep learning because you have to figure out where you're going to insert this in the deep learning network. There are ways of doing that also. I would say not standardized ways, but there are approaches to this with deep learning as well.
Great. Well, thank you so much for that, Ilya. We really appreciate you speaking today. Thank you so much, everyone, for joining us. Again, if you need to contact us, info@viqi.org, or visit our site, viqi.org. And please, 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 we would love your feedback. Take the 1 minute for the survey at the end if you can. I hope you and your families stay safe. Thanks, everyone.
Thank you very much for joining. Take care.